All right, guys, so we are moving on to, oh no, to membranes and proteins. So we were just talking about how proteins are created and some of their functions. Now we're going to learn how they work inside the membranes of our cells and about the membranes themselves. So first thing, cell membranes are going to be a topic we're going to learn about. We're going to get through about half of that in this video. And then transport proteins, signaling proteins, and enzymatic proteins. So a couple different kinds of proteins. So cell membranes first. All right, so a membrane refers to a thin film-like structure that separates two fluids. It acts as a container for the biological system surrounding your probionts, your cells, and your organelles. The video below, which I will actually attach for you, will show experiments done in the laboratory in France to study the properties of liquid. The only substance they used in making this was dye, water, and lipids. The lipids were mixed together and then injected with a solution, mixed together with the dye and then injected in an aqueous solution. And so as you watch the video, I want you to think about some of the properties of the liquids that made it useful. All right. So phospholipids. The membranes of your cells are made of phospholipids, and we learned about phospholipids when we learned about lipids. Phospholipids are amphiphilic, remember, and that amphiphilic has that hydrophilic head and the hydrophobic tail. Now, they are also a double layer because they line up where the the hydrophilic regions are facing outward and the hydrophobic regions are facing inward. And they make the sphere pretty naturally. They kind of started out like a bilayer sheet, they started out like a micelle, and then water would want to come in and get pushed in here, and then it would push it out, and eventually you got this bilayer sheet that it was protected both ways. So the phospholipid forms two parallel lines with hydrophobic ends in between. The hydrophobic ends are protected from the water by the hydrophilic ends, creating that bilayer. Also embedded in there are these cholesterols. These are cholesterols. Those cholesterols kind of keep the membrane more fluid so it doesn't freeze into place. These are good cholesterols that are important in your body. We all know bad cholesterol can clog up your arteries, but these are good cholesterols. So it also makes the bilayer more stable. So your cell membranes are known as a lipid bilayer. Lipids because it's made out of phospholipids, and bilayer because there are two layers. So what's going to happen here is water is going to be attracted to the heads of these phospholipids, and then a new water is going to come in and kind of push at it, and eventually water is going to get pushed to the center. But this is hostile territory. They don't want to be there. So what's going to happen instead is they are going to keep going through until they are out to the other hydrophilic side in the inside of the cell, or vice versa at the outside of the cell. But it doesn't get stuck here in the hydrophobic region, because if it gets stuck there, the cell membrane would actually lyse and open up. So, and then again, the cholesterol kind of immobilizes it and keeps everything a little more stable. It keeps water from going in places it doesn't need to go in, and keeps things a little more put together and, again, fluid. All right. The cell membrane is selectively permeable. What selectively permeable means is that it allows some things in and keeps some things out. It is like a bouncer at a club. It decides what's desirable and what's not. So the phospholipid bilayer will dictate what actually comes through. So here is a picture, another view of a phospholipid bilayer. You get these tightly packed sections. The polar regions are on the outside and with the red, and the nonpolar regions are in the center. But they're not fixed. They kind of float on each other. I picture this like a water, if you had a kiddie pool and you had ball pit balls floating on top, and then also some larger balls floating within there. The larger balls would be your proteins that are going to act as channels and signaling proteins and things like that. And the ball pit balls would act as like the phospholipid heads. So they will create a sheet if there's enough of them, which there is. They'll create a sheet, but they can move past each other. And that is a key component of the lipid bilayer, is that it has a fluidness to it. So there are some small gaps and some tiny things can move through there, but they do want to stick together pretty closely and they can move past each other. So what can pass through just the phospholipid bilayer? What do you think? Polar, nonpolar, small, large? What do you think? Let's see. Do you think O2 will pass through a lipid bilayer? It will pass because it is neutral, so it is nonpolar, and it's very small, only 2 atoms, so it can fit through those tiny gaps, and it will be attracted towards the center and then get pushed all the way through. Good. All right, so the next one. Water, what do you think? Pass or not pass? Ooh, sorry. Water will also pass through. Even though there is a bit of a charge, it's only a partial charge. It's, a polar, but it's not ionic. And because of that, then it'll also move through. And it is also extremely small, just a 3-atom molecule. Okay. How about sodium ion? Yes or no? Sodium is very small. It's 1 atom. However, it has a full charge. Because of that full charge, the hydrophobic region, the middle region, doesn't want anything to do with it, and it will send it away, and therefore it can't go all the way through. How about sugar, glucose, C6H12O6? Pass or not pass? Ask yourself, is it charged, and is it large? It is not charged, so that alone would mean it could pass. However, it is a very large molecule, so it won't fit through those small gaps. So to recap, selectively permeable. The large molecules or charged molecules will not make it all the way through the bilayer. Sugars, ions, nucleic acids, proteins, those things need something to allow them to get through, because the cell does still need the sugars, the proteins, the nucleic acids, but it needs to have a doorway for them to come through rather than just fitting all the way through. So hydrophobic molecules will go through. The small uncharged polar molecules will also go through. The large uncharged molecules will not, and the ions will not. So how do cells get what they need? They, again, are going to need some sort of protein or channel to allow them to open. So the proteins floating in this lipid bilayer are going to can act as channels or signaling proteins depending on which one you're looking at. And again, I was calling these like the larger balls in my ball pit model. And they just kind of bob along. They can go anywhere they want within that. They just, but it'll still maintain a nice cover. So it's a fluid mosaic model because these things can all move around each other, but they still make this nice color, nice solid layer. But because they move around, they're fluid, and they also makes this like mosaic-esque pattern, which is where they get that name from. So proteins will regulate what is in a cell. So proteins are these long chains of amino acids. We learned this last time. They make really useful biological systems. Here is a ribbon diagram of an amino acid chain to make a channel protein. So what you can see is you have your helixes and you have your beta sheets, and you have, it's the quaternary structure, so it's bent on itself with other parts. But you can see that it kind of creates an opening right here. This is what's going to allow things to come through. So there are 2 types of proteins in the membrane. There are peripheral proteins and there are integral proteins. Peripheral proteins are on the sides. They don't go all the way through, so they're just going to be like here. Integral proteins will go all the way through. They are a full channel. So the proteins in the plasma membrane can drift within the bilayer. They are much larger than the lipids, and they move very slowly and make that, again, mosaic-esque pattern. So here is an integral protein, Attached to it is a glycoprotein. It's a carbohydrate that is attached to the protein, and it adds as a point of attachment for other things, cells, bacteria, hormones. It's a signal, it's an attachment area. The glycolipids are going to be attached to the actual membrane, and their purpose is to provide energy for the cellular recognition and act in cellular recognition. So it gives energy to the cell and says, hey, come on over. So the proteins regulate what is in a cell, and an integral protein will make a pore. You can see the pore here, and allow a specific substance, whatever the glycoprotein will signal for, to come through. Even if they're large, even if they have a charge, they can come through because this door is meant for them. So I'm going to also put the links for these videos on here so you can watch these videos and how they move through, and we will go from there. So when diffusion has occurred, there is no longer a concentration gradient. Then blank has been reached. Equilibrium, selectively permeable, phospholipid bilayer, or homeostasis. Get your answer? Did you say equilibrium? You are correct. They're going to want to have equal amounts of something in and out if it's diffusion, which is a passive transport. In osmosis, water molecules diffuse from the plasma membrane into the outside only, outside the plasma membrane to the inside only, from areas of high solute concentration to areas of low solute concentration, or from areas of low solute concentration to areas of high solute concentration. So osmosis, remembering way back from biology, is a passive transport, so it does not require energy. How does it move? Did you say D? From an area of high concentration to an area of low concentration? Oh, wait. Did that say wrong? Oh, that is a typo. It is for sure C. No. Oh, poop. Okay, so in osmosis, water molecules diffuse from an inside plasma membrane to an outside plasma membrane to inside only, from areas of high solute concentration to areas of low solute concentration, and from areas of low solute concentration to areas of high solute concentration. Okay, so osmosis is a passive transport. So the water itself is going to move from a high concentration to a low concentration of water, which is a solvent, not the solute. The solute is going to be these dissolved particles in it, the sugars and stuff like that. If there's more of that stuff dissolved in it, water is going to rush to dilute it. So it's going to go from an area of low solute concentration to where there's a whole lot more to dilute it. So the best answer is going to be D, which might have been opposite of what you were originally thinking, but the word solute is very important in that case. Portion. Good. Okay. What type of environment has a higher concentration of solutes outside the plasma membrane than the inside the plasma membrane? So working with this, is there more, if there's more solute, more stuff, more sugar and stuff outside of the cell versus inside of the cell, is that cell hypertonic, isotonic, normal, or hypotonic? Take a second to get your answer. Did you say hypertonic? You're correct. So in a hypertonic cell, there's more water on the inside and more solutes on the outside, and the water is going to leave the cell to get to that higher concentration. When that happens, the cell will shrivel. So when you put a cell in a hypertonic solution, it will shrivel. What type of solute has a greater flow of water to the inside of the plasma membrane? So hypertonic, isotonic, normal, or hypotonic? Get your answer. It is going to be hypotonic. So now there's way more solutes inside the cell, so water is really flowing in to dilute it. When that happens, the cell itself swells, and so when a cell swells, it's in a hypotonic solution. This is going to be when you water a plant. The plant cells are in a hypotonic environment, so therefore they absorb extra water, and then the plant sits up more because each cell wall is a little more turgid because of the extra water inside that cell membrane. A red blood cell will lyse, which means explode, when it is placed in which kind of solution? Hypertonic, hypotonic, isotonic, or any of these? Get your answer. Sorry. Did you say B, hypotonic? Then you are correct, because water, there's a lot more stuff in that cell membrane, or in that blood cell, so water is coming in, and it's going to get larger and larger, and if it's too much, it will explode. Explode is not quite, it breaks, it lyses, it comes apart, but I always like to think of it as exploding because it makes me happy. All right, dialysis tubing is permeable and to monosaccharides only, so small sugars can get through this dialysis tubing. So which solutes would exhibit a net diffusion out of the cell? So if you look at this picture, let me move my picture, you can see the sugars on the outside versus the sugar on the inside of the cell. And you have sucrose, and glucose. And sucrose, glucose, and fructose. Now ask yourself, what of these are monosaccharides? Is it sucrose is monosaccharide? Nope, it's a disaccharide. Glucose is a monosaccharide, so that's going to be able to move. Sucrose again is a disaccharide, so or a polysaccharide, so it is not going to go through. But glucose and fructose are both monosaccharides, so those can move. So we are going to focus on the sucrose concentrations. This is 0.02 on the inside and 0.01 on the outside. So is the water going to want to go in or leave? Water's going to want to go in. Now, the sugars themselves, which ones will move? Which ones did we decide were monosaccharides? Did you say B? You said B because glucose can move and it's moving out. The question here was what will move out of the cell? Fructose, even though it is a monosaccharide, will not move out of the cell because it wasn't in the cell in the first place. It's already out of the cell. So that's the difference there. All right, the solution outside of a cell, isotonic, hypotonic, or hypertonic. So let's look at this again. We have 0.01 molar of sucrose, glucose, and fructose, and we have 0.05 and 0.02 on the inside. So is the cell itself, is the solution outside of the cell isotonic, hypotonic, or hypertonic? Is water going to come in or out? Did you say water was going to come in? Then the cell is hypotonic, and it has the possibility of lysing if it doesn't stretch enough. Good job. All right, the process by which a cell ingests large solid particles is known as cell eating. So that term is going to be pinocytosis, phagocytosis, exocytosis, or osmoregulation. Again, this is all the way back to Bio 1. Take a minute, get your answer. Did you say phagocytosis? You are correct. Phagocytosis is cell eating. It's going to engulf large things. Pinocytosis is cell drinking. It's going to kind of take in small things. And they're both taking in, so they're endocytosis, not exocytosis. All right, antibodies are embedded in the cell membrane and bind to antigens on the surface of foreign cells. What type of molecule is an antibody? So it is embedded in the cell membrane. Is it a phospholipid, a glycolipid, a glycoprotein, or an enzyme? Did you say C, glycolipid? You are correct, because it is not attached to a protein. It is in the cell membrane, so it is a glycolipid. And that is where we're going to stop today. So we talked about how things move in and out of the cell, osmoregulation. We talked about hypotonic, which is a cell in a hypotonic solution will increase in size, maybe lyse. We talked about hypertonic solutions. If a cell is inside a hypertonic solution, the water is going to leave and therefore it will shrivel. And isotonic we didn't talk too much about, but isotonic is going to be equal on both sides. If it's equal on both sides, then net water is going to move in and out at the same rate, so it's just going to be nice and equal. It's not going to change. We also talked about membrane proteins and the fact that large and charged things can go through those. We talked about the cell membrane itself, its fluid mosaic model, and how nonpolar and small things can move through the lipid bilayer just through the tiny gaps. And that is it for today. All right, I will talk to you guys later.